Next up, we have Ayaan Ismail. He's a 14-year-old nanotech expert who will be talking about the future of healthcare treatment, personalized medication, and his startup that he co-created to help society better understand their own genetic information for better long-term health. This summer, he worked at a nanotech lab printing functionalized nanomaterials and sensors. Please welcome him to the stage. Awesome.
A few months back, my mom fell ill with pneumonia. And so she went to the doctor, and the doctor prescribed her with both a puffer and some antibiotics. And so she took both of these medications for the week, and after the week she realized that her condition started to worsen instead of get better. And so she went back to the doctor, and the same cycle repeated for the second medication and the third medication. Until finally, on the fourth medication, did her condition actually start to get better. And so I researched deeper, and I realized that this is actually a humongous problem, and that about 22,000 people die each year due to the fact that the medication they are given is toxic towards them. And so I wondered, why does this occur? And so I researched even deeper, and I realized that everyone responds differently to a medication. So while you might have a toxic response to a drug, I might have a normal response, and someone else might have no response at all to a medication. Now this blew my mind, as I realized that what we're doing with medication today is just plain out wrong, and that we should be personalizing it to individuals' genetic code and metabolizer. And so this is actually the study of pharmacogenomics, in which we look at the correlation between a patient's genetic information and their metabolizer to be able to devise a medication that has optimal effectiveness on them the first time, so that they don't have to keep on coming back to the doctor. Now, before we dive a little deeper, let's just take a step back and look at the basics. Every living organism has cells, and inside these cells there's a nucleus. This nucleus holds chromosomes, and these chromosomes hold our DNA, the cookbook to our life, the essential genetic information that codes for how we react to certain situations and how we look. Now, not only do our chromosomes hold our DNA, they also hold alleles. And alleles are mutations found on the same spot on the chromosome that code for visual characteristics of an organism that we can see. So in this case, the color of the flower. Now, not only do small mutations, such as a small a or a small t, code for why you might have blue eyes and why I have brown eyes, but also for why medication might have optimal effectiveness on you in comparison to me. And so let's just say someone's taking codeine for pain relief. Now the active form of that's morphine. And let's just say person A has allele 1 from father and allele 1 from mother, coding for the CYP2D6 allele 1 gene. Now they also have a normal metabolizer, which means that they metabolize the medication as any normal person would. And so therefore, we would give them the normal dosage of a medication with normal toxicity. And so we would give them codeine over morphine. Whereas person B has allele 2 from father, and they also have an ultra-rapid metabolizer, which means that they metabolize the medication much more rapidly than a regular patient would. And so instead of giving them a high toxicity and high dosage of codeine, we would just give them morphine instead. And so with all my knowledge in pharmacogenomics, I decided to start my own experiment, in which I personalized certain blood clot medications to specific genotypes. More specifically, I personalized aspirin, warfarin, and clopidogrel to genotypes AA, AG, NA, and AG, as well as GG. Now there are 3 main steps in my process, the first of which I put certain genes and enzymes in a Lumini simulator. What I came out with looks something like this. But what I specifically want to point out is that we're looking at the VKORC1 gene. And so now that we know what genome we're looking at, the next thing that we have to figure out is the variant associated with our genotype and blood clot medication. So in this case, genotype AG and aspirin. And what we're looking at here is a genome browser, which encompasses all of the genetic information on our specific genome. what I want to point out is that we're looking on the left side of the chromosome, indicated by the vertical red bar, and as we look at different variants, this will change. I've already put in the variant number, but this is actually very hard to find, but with a little bit of digging, I was able to tell that this is the variant in correlation with our genotype and medication. And so the next thing that we have to figure out is how does the drug actually get metabolized in the body? And what's the end result out of all of this? What we're looking at here is the specific genome associated with our variant. And in that case, that's the ABCB1 gene. Now, while this might look complicated, that's because it is complicated. And what I specifically want to point out is that these bars represent the steps in the metabolic process. So for example, the first two orange bars show how when the drug is first inhibited, it calls on calcium to decay at the platelet, which is what's causing the blood clot. But then, indicated by the high green bar, we can see that we've called on receptors and proteins. And so overall, we can conclude that genotype GG is best with clopidogrel, whereas genotype AA, AG, NA, and AG is best with aspirin and warfarin. Now, not only can this process be done for blood clot medications, but also for pneumonia drugs, cancer drugs, and even deep vein thrombosis drugs. And so the question is, if the technology is there, then why aren't we doing it? But wait, that's the problem. The technology just isn't there. The technology that we need to be able to understand our genetic information, to sequence our DNA, is askewed. It can take at least 2 to 3 weeks, and when trying to relieve a patient of their sickness, this is way too long. The effectiveness is lowered as we can miss out on certain variants because the process is just not as efficient as it could be, and it can cost from $500 to $2,000. And let alone, we're not even talking about the cost for variant analysis for the purpose of our genome. And so looking at all these problems in genetic sequencing, me and my co-founder, Samarth, decided to create our own startup, Genis, in which we're leveraging nanotechnology and quantum physics to sequence genes much more effectively than current methods. Our mission is to dramatically improve the accessibility of genetic sequencing to improve the health span of everyone worldwide. We've been able to talk to and gain support from companies such as Johnson & Johnson, PillCheck, which is North America's largest personalized medication company, University of Toronto, University of Waterloo, and many more. To get a sense of what our product looks like, here's our simulation. Indicated in the blue and yellow, we have our silicon nitride nanopore. Now there's a hole in this, and the DNA will pass through this hole. There's an electrical field in this hole, and since each nucleotide in our DNA, A, C, T, and G, give off a different current when in an electrical field, we'll be able to tell what nucleotide it is based on the current it gives off. The DNA is shown in the green and purple double helix structure, which is what's passed through the nanopore. Now with Genis, we're able to more time effectively, more cost effectively, and much more efficiently sequence the genome. Not only can we create personalized medications, we can also do things like disease and cancer detection and look at certain mutations in a genome that might allow it to become cancerous. We can see if someone's genetics are right for a space environment and what might happen to their bones when they're in a zero-gravity environment as well. We can look at gene editing technologies such as CRISPR and see the off-target effects of them and try to make these editing technologies as amazing as they possibly can be. We can leverage this data for AI and put it through models and try to look at the variants to try to improve the human overall. We can leverage things like gene therapy and insert new DNA into the cells so that there will be no bad mutations and we won't get a sickness in the first place. And we can create personalized medications. And so if there's one thing you're to take away from this presentation, it's that our DNA is key. Our genetic information holds the key to a lot of mysteries we currently do not know about ourselves. And when we look back at this in the next 10, 20, 30 years, we'll see how insane it was that we were giving everyone the same red pill and how we weren't leveraging genetic sequencing to be able to give everyone personalized medication. With Genis, we'll be able to more time effectively, more cost effectively, and much more efficiently sequence the genome to unlock the power of the genome and solve some of the world's biggest problems. Thank you.
So, besides like going home and throwing out all your medicine and like ordering a 23andMe kit, you have questions for our friend here?
Do you envision any pushback from the pharmaceutical industry with having to create individualized medication on a sort of case-by-case basis as opposed to creating more widespread, easily usable, maybe not easy, more widespread use medication?
Yeah, so I think it'll be a setback for pharmacies, but for overall as humanity, it's going to improve us because the medication we'll be taking will be at optimal effectiveness and allow us to just not get sick, and we won't have to keep on going back to the doctor if the medication isn't effective.
Any other questions? Humanity 1, Pharmacy 0.
Well, currently with Genis, we're working on in a lab to create our prototype. So that will be done soon, and then we can leverage that data to create things like personalized medicine, but I'd say in the next 10 years. Companies are already starting to sequence genes. Companies like Manulife, like even big companies that are not even in healthcare, are providing like their employees with genetic sequencing as well. So you're starting to see it become more integrated into society as well.